Welcome back to the Deep Dive. We're here again to unpack complex ideas, hopefully give you some fascinating insights, and really help you get well informed. Today, we're diving deep right into the absolute command center of, well, pretty much every eukaryotic cell, the nucleus. We're drawing our insights from Histology. Texan Atlas 8th edition is a fantastic resource, really helps visualize this microscopic world, and it connects structure to function and, ultimately to health and disease. So our mission today, discover how this tiny, often overlooked organelle really orchestrates the entire symphony of life and death within us.
It's definitely an incredible mission. When we look at the cell nucleus, we're basically looking at the brain of the cell. It holds all the genetic blueprints, right? All the sophisticated machinery needed for life's most essential processes. Things like DNA replication, RNA transcription, all that key stuff. And what's truly remarkable, thinking about that microscopic anatomy, is how much we can actually tell about a cell's health, its activity, just by looking at its nucleus. It's like a tiny but really powerful diagnostic window.
Okay, let's unpack this then, starting with the foundational overview. So, at its core, the nucleus is this membrane-limited compartment. Its main job is to safely keep the cell's entire genetic library. And in a non-dividing cell, what we call an interphase cell, you find these key components inside. There's chromatin, the nucleolus, the nuclear envelope, and then just the general nuclear fluid, the nucleoplasm.
Right, and that microscopic evaluation you mentioned, it's absolutely crucial, especially in fields like, say, cancer diagnosis. Even slight changes in nuclear size or shape or the structure inside, they can be really significant indicators. For instance, when cells are dying, they go through these very specific visible changes that histologists look for. There's pydornosis, that's where the genetic material, the chromatin, it condenses down, the nucleus shrinks into this dense, dark blob. Then there's karyorrhexis, that's where the condensed nucleus actually fragments, breaks apart. And finally, karyolysis, where the DNA just dissolves completely and the nucleus sort of fades away. Seeing these changes tells us a huge amount about the cell's state if it's distressed.
It's truly incredible how much information is packed into such a tiny space. And speaking of packing, chromatin, get this, each of your cells has about 1.8 meters of DNA stretched out. That's, 100,000 times longer than the nucleus itself? How on earth does it all fit?
Yeah, it's amazing. The DNA has to be incredibly folded, really tightly packed. And this is done by forming chromatin, which is basically a complex of DNA wrapped around these special proteins called histones. This complex structure is actually what gives the nucleus its characteristic basophilia. That means it stains easily with basic dyes, making it really stand out under the microscope in histology slides.
Okay, so it's DNA plus proteins packed tight.
Exactly. And it's an ingenious system of organization. The first level of folding. The DNA coils around a core of 8 histone molecules. This creates what we call nucleosomes. You often see them drawn as beads on a string.
Yeah, I know that image.
Right. Then those coil up further into denser structures like 30-nanometer fibrils, and then into larger loop domains anchored to a kind of internal scaffold. And within the nucleus, chromatin mainly exists in 2 forms. These tell you a lot about what the cell's up to. First, there's heterochromatin. That's the tightly condensed, densely staining form. Think of it as the cell's archive genes here are mostly inactive, not being read. You often see it clumped at the edge of the nucleus or near the nucleolus. Cells that aren't very active, like small circulating lymphocytes, they tend to have a lot of this.
Okay, so that's the stored inactive stuff. What's the other form?
The other is euchromatin. This is the dispersed, lightly staining form. This is where most of the transcriptionally active genes are located, the open books the cell is actively reading from. It's much more prominent in cells that are really busy metabolically active, like neurons or liver cells. You can see the difference clearly in stained sections.
Okay, wow. And this is where it gets really interesting, connecting this to the bigger picture. Think of the Human Genome Project. Back in 2003, it mapped, 2.85 billion base pairs, identified around 23,000 protein-coding genes. But one of the really surprising things I remember was the discovery of copy number variations, CNVs.
Oh, absolutely. That was a huge deal.
Yeah. We kind of assumed most genes came in 2 copies, one from each parent. But CNVs showed, the number of copies can actually vary quite a bit, sometimes 1, sometimes 3, maybe more. And this wasn't just a minor detail. It showed that our individual genetic landscapes are much more dynamic than we thought. Helps explain differences in susceptibility or resilience to diseases. It really forced us to sort of rethink the definition of a gene itself.
It certainly did.
And when cells get ready to divide, all this chromatin condenses even further into those distinct structures we recognize as chromosomes. Clinically, Those protective ends, the telomeres, they're super important, right? They shorten each time a cell divides, acting like a kind of cellular clock.
That's right. And it's significant in cancer because many malignant cells manage to switch on an enzyme called telomerase. This enzyme rebuilds the telomeres, basically making the cells immortal, allowing them to keep dividing indefinitely. That's a key feature of cancer, and telomerase is a potential target for therapies.
Right, a way to stop them dividing.
Exactly. And analyzing these chromosomes using karyotypes is a fundamental diagnostic tool. We can use techniques like FIH fluorescent in situ hybridization to actually visualize specific bits of chromosomes. This lets us spot abnormalities like translocations, where parts of chromosomes swap places, like between chromosome 8 and 14 in some lymphomas, or we can see deletions, missing pieces, like in Prader-Willi syndrome.
Wow.
And a really cool visible example of this chromatin state difference is the bar body. You find this small, dense spot in the nucleus of female cells. It's actually an entire X chromosome that's been inactivated, a type of heterochromatin. Its presence or absence can even be used for prenatal sex determination just by looking at cells.
Fascinating. Okay, let's move to another key nuclear component, the nucleolus. You described it as this non-membranous region inside the nucleus, and it's basically the cell's ribosome factory.
That's its primary job, yes. It's the site where ribosomal RNA, or rRNA, is synthesized, and where the initial assembly of ribosomal subunits begins. Microscopically, you can often distinguish different regions within it. There are fibrillar centers containing the rRNA genes themselves, then the fibrillar material, where those genes are actively being transcribed into RNA, and finally the granular material, which is where those previsomal particles start to get assembled.
So it builds the parts for the protein factories.
Pretty much. Once partially assembled, these ribosomal subunits are shipped out of the nucleus through the nuclear pores into the cytoplasm for the final steps of maturation. And it's more than just a ribosome factory. We now know the nucleolus is involved in other things too, like helping regulate the cell cycle. And some viruses can actually hijack the nucleolus for their own replication. It's a busy place.
Okay, surrounding all of this vital machinery is the nuclear envelope. You said it's a double membrane system, separating the nucleus from the cytoplasm. And what's really neat microscopically is that its outer membrane is continuous with the rough ER, right? Part of a larger network.
Exactly. It links the nucleus directly into the cell's protein synthesis and modification pathways. And supporting the inner nuclear membrane is something crucial, the nuclear lamina. Think of it like a meshwork, a scaffold made of proteins called lamins, which are intermediate filaments. It's not just for structure, though. It's vital for DNA replication, gene regulation, and just organizing the chromatin inside. And uniquely, this lamina disassembles during mitosis, allowing the chromosomes to separate, and then it reassembles afterwards.
Wow, it breaks down and rebuilds itself.
Every time the cell divides, yes. And problems with these lamin proteins, caused by genetic mutations, lead to a group of diseases called laminopathies. Things like certain types of muscular dystrophy, like Emery-Dreifuss.
So defects in this nuclear scaffold cause major diseases.
Okay. And what about getting things in and out? The envelope isn't solid. You mentioned pores.
Right, nuclear pores. These aren't just simple holes. They are massive, intricate structures called nuclear pore complexes, or NPCs. Yeah. Each NPC is like a sophisticated molecular gatekeeper, an octagonal structure with fibrils reaching into the cytoplasm and a basket structure on the nuclear side. It's absolutely vital because it controls everything that goes between the nucleus and the cytoplasm. Small things like ions and water can diffuse through pretty easily, but larger molecules, like proteins that need to work inside the nucleus or RNA that needs to get out, they need specific passcodes, nuclear localization signals, or nuclear export signals to get transported through the NPC. It's tightly regulated.
A very selective doorway. Extremely selective. And the stuff filling the rest of the nucleus, besides the chromatin and nucleolus, that's the nucleoplasm, it used to look kind of empty or amorphous in older microscopic images, but we know now it's a rich, dynamic soup. It's packed with essential proteins, metabolites, ions, and all the machinery needed for things like gene transcription. It's where a lot of the action happens.
Okay, so we've got this incredibly complex command center, the nucleus, housing all the genetic info machinery, but it's not just static, right?
Yeah.
It's about action, growth, division, even self-destruction, that whole cycle of cellular life. Let's shift gears now and talk about cell renewal. Cells are constantly managing their populations. How do we categorize them?
We can broadly group cell populations based on their mitotic activity, their tendency to divide. First, you have static cell populations. These are cells that basically don't divide once they're mature. Think of nerve cells or cardiac muscle cells. They're terminally differentiated. Then there are stable cell populations. These normally divide quite slowly, but they can ramp up division significantly if there's an injury or a need for repair, like liver cells or smooth muscle cells. And finally, you have the renewing cell populations. These divide regularly to replace cells that are constantly being lost or worn out. Blood cells, skin cells, the lining of your gut, these are always turning over, some rapidly, some a bit more slowly.
So constantly dividing, or dividing when needed, or basically never dividing.
That's a good summary. And this whole process is governed by the cell cycle. It's a tightly regulated sequence of events controlling cell growth and division with the main goal of producing two genetically identical daughter cells. The cycle has two main phases. Interphase, that's the growth and preparation period, and M phase, which is mitosis, the actual division part. Interphase is further broken down into G1, S, and G2 phases. And critically, throughout this whole cycle, there are internal quality control points called checkpoints. These act like molecular gatekeepers, making sure everything is correct before the cell proceeds to the next stage. They can halt the cycle if there's damage or something's wrong.
Okay, let's walk through interphase then. G1.
G1, or gap 1, is usually the longest and most variable phase. This is where the cell grows, gathers nutrients, and synthesizes the RNA and proteins it needs for the next steps. It contains a really crucial checkpoint called the restriction checkpoint. You can think of it as a point of no return. Here, the cell assesses conditions. Is it big enough? Are there enough nutrients? Are the signals right? And decides whether to commit to division by entering the S phase or to exit the cycle and enter a resting state called G0, maybe permanently. There's also a key DNA damage checkpoint in G1. If DNA damage is detected, a protein called P53 becomes active. If the damage is too severe to repair, P53 can trigger apoptosis programmed cell death, a crucial safeguard.
A point of no return and a safety check. What happens next in S phase?
S stands for synthesis. This is where the cell replicates its entire DNA content. Each chromosome is duplicated, forming two identical sister chromatids joined together. And of course, there's another checkpoint here, the S DNA damage checkpoint, monitoring the replication process.
Got it. DNA copied, then G2.
G2, or gap 2, is a shorter phase where the cell prepares for mitosis. It finishes growing, reorganizes some organelles, and importantly, double-checks the replicated DNA for any errors or damage. So there are more checkpoints here. The G2 DNA damage checkpoint and the unreplicated DNA checkpoint, ensuring everything is perfect before the chromosomes try to segregate in mitosis.
Okay, these checkpoints sound absolutely critical. What happens if they mess up?
Well, that can lead to what's called mitotic catastrophe. If a cell with significant damage or errors manages to bypass these checkpoints and enters mitosis anyway, it often results in abnormal chromosome segregation, leading to daughter cells with the wrong number of chromosomes aneuploidy. This is a major mechanism contributing to the development of cancer. Many malignant cells have faulty checkpoints. For example, cancer cells often lose contact inhibition. Normal cells stop dividing when they get crowded, but cancer cells just keep going. This is often linked to problems with that G1 restriction checkpoint we mentioned.
So loss of control equals uncontrolled growth.
Essentially, yes. Certain viruses, for instance, produce proteins that can actually disable key checkpoint regulators like P53. Making the cell cycle controls ineffective.
Okay, so who's actually driving this cycle forward, step by step?
The engine of the cell cycle involves protein complexes that appear and disappear in a cyclical fashion. The key players are proteins called cyclins, whose levels rise and fall throughout the cycle, and enzymes called cyclin-dependent kinases or CDKs. CDKs are always present, but they're only active when bound to a specific cyclin partner. The discovery of the first major complex, initially called maturation promoting factor, or MPF, which turned out to be a combination of CDK1 and cyclin B, was a huge breakthrough. It acts as the master switch to trigger entry into mitosis.
So cyclins build up, activate a kinase, push the cell to the next phase, then the cyclin gets destroyed.
That's the basic idea. Different cyclin CDK complexes control different transitions, G1 to S, S phase progression, G2 to M. Their activity rises sharply when needed and then drops off, often because the cyclin component is rapidly degraded. And there are also specific inhibitor proteins that can block cyclin CDK activity, providing another layer of control, especially at the checkpoints. It's a very intricate regulatory network.
Okay, intricate control. Now let's get to the main event. Mitosis. This is the actual process of chromosome segregation and nuclear division, producing two genetically identical daughter cells, right? Same chromosome number, same DNA content as the parent.
Exactly. Mitosis follows the S phase, where DNA was doubled, so cells entering mitosis actually have double the normal DNA content temporarily. We call it fourpetal. The goal of mitosis is to accurately divide that duplicated material into two new nuclei.
And how does it happen? What are the stages?
Mitosis is usually divided into four key phases, though it's a continuous process. First is prophase. Here, the replicated chromosomes condense and become visible under a light microscope. The nuclear envelope starts to break down, the nucleolus disappears, and the mitotic spindle starts to form between two centrosomes that move to opposite poles of the cell. Special structures called kinetochores assemble on each chromosome's centromere. These are like handles for the spindle microtubules to grab onto.
Okay, chromosomes condense, envelope disappears, spindle forms.
Then comes metaphase. The mitotic spindle is fully formed now, made of different types of microtubules. The chromosomes, attached to spindle microtubules via their kinetochores, are maneuvered until they all line up precisely along the equator of the cell, forming the metaphase plate. This alignment is crucial for equal division.
Lined up in the middle, ready to split.
Right, which happens in anaphase. The proteins holding the sister chromatids together suddenly break down. The sister chromatids separate, becoming individual chromosomes, and are pulled rapidly towards opposite poles of the cell by the shortening kinetochore microtubules, powered by molecular motors. The cell elongates.
They separate and move apart. What's the final stage?
Telophase. The separated chromosomes arrive at the poles and begin to decondense, becoming less visible. A new nuclear envelope reforms around each set of chromosomes, creating two distinct nuclei. The nucleoli also reappear, and usually overlapping with late anaphase and telophase is cytokinesis, the division of the cytoplasm. A contractile ring made of actin and myosin filaments forms around the middle of the cell and pinches it in two, creating two separate daughter cells, each with its own nucleus containing an identical set of chromosomes.
Wow, a very precise dance, ensuring exact copies. Now let's contrast that with meiosis. You said this is different. It produces gamete, sperm, and egg with half the number of chromosomes in DNA. Why is that important?
It's absolutely essential for sexual reproduction. If gametes had the full set of chromosomes, like somatic cells, then the chromosome number would double with every generation after fertilization. Meiosis reduces the chromosome number by half, from diploid 2n to haploid 1n, so that when sperm 1n fertilizes egg 1n, the resulting zygote restores the normal diploid number 2n. Equally important, meiosis generates incredible genetic diversity through two main mechanisms, crossing over an independent assortment of chromosomes.
Okay, so half the chromosomes and genetic shuffling. How does it achieve this?
Meiosis involves two sequential nuclear divisions, meiosis of the first and meiosis the second. But crucially, there's only one round of DNA replication before meiosis first. No S phase between the two divisions. Meiosis first is often called the reductional division because it separates homologous chromosomes. Prophase is very long and complex. Homologous chromosomes, one inherited from each parent, pair up precisely in a process called synapsis, forming structures called bivalence. While paired, they physically exchange segments of DNA. This is crossing over or recombination. It shuffles alleles between maternal and paternal chromosomes. Then in anaphase down, it's the homologous chromosomes that separate and move to opposite poles. Importantly, the sister chromatids remain attached at their centromeres. So the cells resulting from meiosis are haploid in terms of chromosome number 1, but each chromosome still consists of two chromatids, so the DNA content is still technically diploid 2p.
Okay, so homologous pairs split in the first division, then meiosis the second.
Meiosis the second is much more like a standard mitotic division. It's called the equatorial division. The chromosomes line up at the equator again in metaphase 2. Then, in anaphase 2, the sister chromatids finally separate and move to opposite poles. This results in 4 cells, each with a haploid chromosome number 1n and a haploid DNA content 1d. And because of crossing over in meiosis the first and the random way homologous chromosomes lined up and separated, each of these 4 resulting gametes is genetically unique.
So reduction division first, then splitting the copies, leads to unique haploid gametes.
Exactly. Although there's a difference in outcome between males and females. In males, meiosis produces 4 functional spermatids. In females, cytokinesis is unequal, producing 1 large ovum egg and 3 small, non-functional polar bodies.
Right. Okay, so we've covered division. What about the end of the line, cell death? You mentioned it's not always bad, it's actually vital.
Absolutely vital. Maintaining homeostasis, that steady state in the body, requires a careful balance between cell proliferation, making new cells, and cell death, removing old or damaged ones. If you have too much cell division or too little cell death, you can get uncontrolled growth, like cancer. Conversely, if you have too much cell death or not enough replacement, you can get degenerative diseases like Alzheimer's or Parkinson's, where crucial cells are lost.
So it's a critical balancing act. What are the main ways cells die?
There are two major pathways historically described. The first is necrosis. This is usually considered accidental cell death, typically caused by acute injury like toxins, lack of oxygen, or physical trauma. It's a messy process. The cell swells up rapidly, its membranes rupture, and all its internal contents spill out into the surrounding tissue. This release of cellular debris triggers a significant inflammatory response, attracting immune cells to clean up the damage. Think of the redness and swelling around a wound.
Okay, messy, accidental death with inflammation. What's the alternative?
The alternative is apoptosis. This is often called programmed cell death or cellular suicide. It's a highly regulated physiological process where the cell actively orchestrates its own destruction in a clean, controlled way, usually without causing inflammation. Apoptosis has very distinct features. The cell actually shrinks, its DNA gets systematically chopped up into fragments, and its mitochondria become leaky. Leaky mitochondria release proteins like cytochrome C, which activate a cascade of enzymes called caspases. These are the executioners, the cell's internal suicide squad. The cell membrane starts blebbing, forming little bulges, and eventually the cell breaks apart into small membrane-enclosed fragments called apoptotic bodies. These apoptotic bodies are quickly engulfed and digested by neighboring cells or phagocytes, so the whole process happens tidily, without spilling contents or alarming the immune system.
Wow. A clean, programmed self-destruction. What triggers it?
It can be triggered by a wide range of signals, both external and internal. External signals might include signals from immune cells, like tumor necrosis factor binding to death receptors on the cell surface, or maybe damage from radiation. Internal triggers could be things like irreparable DNA damage detected by p53, or signals from activated oncogenes suggesting something is wrong with cell growth. In fact, that mitotic catastrophe we talked about when cell division goes badly wrong often triggers apoptosis as a safety mechanism.
So the cell can decide to self-destruct if things go wrong. Can it be stopped?
Yes, there are also survival factors, signals from other cells or the environment, that actively inhibit apoptosis. The decision between life and death is often controlled by a crucial family of proteins called the BCL2 family. Some members promote apoptosis while others inhibit it. The balance between these determines the cell's fate.
A constant push and pull between life and death signals.
Precisely. And, research is showing the picture is even more complex than just necrosis versus apoptosis. There are other fascinating forms of programmed cell death being described. For example, anoikis, that's a type of apoptosis, triggered when a cell becomes detached from its proper surroundings, its neighbors, or the extracellular matrix. It's a key way to prevent cells from growing in the wrong place, like in metastasis. Then there's pyroptosis, which is often triggered by infection and is inherently inflammatory, involving different caspases. And necroptosis, which is fascinating because it's a regulated, programmed form of necrosis using different molecular pathways. What's becoming clear is that these different death pathways aren't always mutually exclusive. They can share components, and sometimes multiple forms of death might happen in a tissue at the same time. It's a really active area of research.
Wow, it's incredibly complex. What a profound journey we've taken today. Really diving right into the cell's nucleus. From seeing how nearly 2 meters of DNA gets meticulously packed into that tiny space, to understanding the precision of mitosis and meiosis ensuring life continues, and then exploring the complex, balanced processes of programmed cell death that maintain our health. It really drives home that understanding these microscopic details, the histology and cell biology, is just fundamental. Fundamental to grasping life itself and understanding the basis of health and, well, disease too.
Absolutely. I think this deep dive really highlights the incredible precision, the delicate balance that sustains life at this most fundamental level. And to think that just small disruptions, tiny glitches in these processes hidden inside the nucleus can have such huge widespread consequences for the entire organism, it really underscores the wonder, doesn't it? The complexity of the body's internal machinery. Hopefully it gets you thinking about what's really going on inside, right down at the cellular level.
It certainly does. A fantastic perspective. Thank you for joining us on this deep dive today. We hope you'll keep exploring the fascinating world both around us and, clearly, deep within us. Until next time.